Hello friends, good morning. I am Professor Priyanka Aire, Department of Microbiology from KANMS College, Satana District, Nasik. Welcome back dear friends to another video lecture on the different topic, that is easiest way to remember cell signaling pathway. Okay, JAK-STAT signaling pathway. It is very fast way to remember any complicated pathway. Just is for you, and actually will be device system can help us to remember and recall cell signaling quite this easiest way. So what calling it has to 5 fingers to remember concept to cell signaling pathway. Okay, and it's nothing but to think about 5 fingers. In this case, if your left or on your left hand at the palm, all the writing is there. Now your 5 finger, right? In this 5 finger, five important parameters of cell signaling, which is will not be justify will be justified. Now whenever you reading a cell signaling pathway, these 5 parameters are the most thing for every single cell signaling pathway. All right, so JAK-STAT signaling pathway, it is a chain of interaction between protein in a cell, and it is involved in process such as immunity, cell division, cell death and tumor formation. Okay, so in this video lecture we are focused on JAK or STAT pathway. That is, JAK-STAT pathway is all about in immunology. So obviously it is linked with the cell signaling, goes inside the immune system, of that is the activation of lymphocytes or T cell getted activated and B cell getted activated, and WBC cells are even forms depends on the JAK-STAT pathway. So definitely JAK-STAT pathway JAK-STAT pathway, in other words, it is a chemokine pathway or cytokine signaling pathway, and that is what is all about. So again, remember and recall, JAK-STAT pathway is one of the most easy way is used to 5 finger concept of cell signaling pathway. And again, as per the 5 finger concept, you can see our fingers. Okay, so let's put the actual name of parameters in this case of JAK-STAT pathway by looking at this 5 finger concept. Very first parameter, that is I will tell you first finger, that is signaling molecule, and we can also see in this diagram, and I am explaining it. We can consider our first finger, which is very first parameter that I will tell you. First finger, that is signaling molecule, and of course the signaling molecule in this case cytokine. Now second important parameter in this receptor, you always know the receptor names, that is easy to remember. This is always linked with the name of signaling molecule. So in this case cytokine receptor, we can also kind of chemokine receptor. There is difference between chemokine and cytokine receptor, discuss that other lecture. Okay, but still it's a cytokine receptor. And the third, we can see in this figure, the third important parameter of JAK-STAT pathway is cellular activator, and the cellular activator in this case are JAK-STAT obviously. So JAK is a protein, so signaling pathway is quite easily. One cytokine binds to receptor get dimer, and it is phosphorylate JAK, and once JAK get phosphorylate, JAK also phosphorylate the cytosolic domain in of the cytokine receptor. We can also see in this diagram. Okay, so that the cellular activation process is actually quite simple to the other cell signaling pathway. Okay, now fourth important parameter, that is we consider your fourth important parameter, that is transcription factor. Okay, the transcription factor in this case the STAT protein dimer. So STAT protein dimer acting as a transcription factor. Remember, whenever JAK get activated phosphorylates and the cytosolic domain of the cytokine receptor. Once the cytokine receptor of cytosolic domain in its phosphorylate, then the STAT protein which is already present in cytosol and it is bind with cytosolic domain of cytosine receptor. Okay, and then they get phosphorylate. Okay, so once the STAT protein get phosphorylate, two such STAT protein are phosphorylate, they are going to combine with one another forms of the dimer. Okay, you call it STAT protein dimer or STAT dimer. Next, once a STAT dimer forms, that STAT dimer goes inside the nucleus and it will bind to the promoter, and it's control the activation of certain genes, transcription of genes downstream. Okay, and what are the we can see in that picture, what are these genes, what are the genes are product actually do the self effects. Okay, different different JAK and different STAT protein, and the combination of different JAK and STAT leads to the activation of different transcriptional activation of different genes. For example, JAK lymph is the link with the function known as a lymphocyte, and the development where JAK is linked with ultimate cellular effect of the interferon gamma signaling, where STAT4 once get activated as a dimer, and transcription factor is causes activation of natural killer cell. While on the other hand, when the STAT protein get dimerized and get activated, goes inside the nucleus, it causes a transcription of the other gene product. Okay, so they are the ultimately leads to the activation and formation of white blood cells. Okay, and there is so many other examples, is just only put for example, but there is so many. Okay, friends, JAK and STAT combination, they have specific event and specific effect to be done. Okay, here I am showing the mechanism of the JAK and STAT pathway. Okay, in that slide, and here we see the structure of JAK and STAT pathway. Friends, JAK and STAT combination, see, this is the pathway of JAK and STAT combination, they have specific event. Okay, and this is a specific effect to be done. So that is regarding JAK and STAT pathway. Remember, whenever in JAK and STAT, so all about JAK activation of phosphorylation in the cytosolic domain of cytokine receptor, STAT acts as a dimer. STAT dimer acts as a transcription factor. Results are very huge, because in immunology all the cytokine signaling actually depends on JAK STAT signaling. So signaling pathway is the same, but signaling components are different, and we change it component, we are going to see different effects impact in the cell. Okay, thank you so much guys. This is a very simple way, that is 5-finger methods or 5 parameters which we will have to recall, and we can signaling components are the we can easily understand the JAK and STAT pathway. Okay, thank you so much.